Now we can move to the next talk from Tim Derby, Introduction to Diffusion Tractography. Tim is from the Danish Research Center for Magnetic Resonance, Copenhagen, Denmark. And up to you.
All right.
Oh, maybe just a quick announcement. Please, if you have time, try go online and do the evaluation for us. It's very important to get feedback, not only about the score, but also if you have a suggestion and advice for how we can improve next year. Thank you.
Okay, thank you. And I'm going to talk about, give you an introduction to diffusion tractography. And now we have two screens. I don't have a pointer, so I have this laser, so I will try to point on this one. So I hope you guys can see what's happening over here. Sorry for that. But tractography, we know that we have this nice spaghetti fields of the whole brain, and that one we is like to try to infer that this is related to the axon network in the brain. And from that one, we actually can try and infer something about how these different brain structures are connected together, for example, cortical to cortical connections. And we can make graph theories and all these smart things to look at the brain connectomics, giving this tractography. We can also look, instead of looking at the whole brain network, we can also look at specific tracks in the brain using tractography. So we can have an example of the IFOF can be extracted here, where you can place some different seed regions and waypoints to extract the tracks. But there's also challenges here. So if you if you go and look at the different subjects, two other subjects, then probably you will not extract the same kind of tracks, even that you use the same seeding procedures. So there's a lot of challenging when you start to work with tractography, how you robustly extract your tracks and do your analysis, and that's really impact your analysis and interpretations afterwards. So, like to be a good tractographer, it's really a matter of training and experience. You need to do this regularly to get experience how you really work to the data set and get into that. And that for sure you will not learn from this lesson. But what I can give you is something, it's really essential to have like insight into how your tractography method works and how is actually the relation to the underlying true axonal networks that you're working with. How is that link? And this is what I will try to cover in this lecture. So I put together some objective learnings here. So first, you will get insight to how does actually the true axonal networks look like. Then we'll look into the basic pipeline of tractography. You heard some of the processing stages today, this morning. And then we will look into the different seeding strategies to extract the tracks. And then, of course, you should get some insight into the different sources of ambiguity that can influence your tracking results. So this is like the outline, which cover the different objectives I was saying. And then in the last part, I will give you an example of what we call anatomical connectivity mapping. It's a new way like to do kind of voxel-wise analysis based on tractography. And you can actually enhance your like biomarker strengths combining with conventional local estimates like the DTI. So let's have a look. How does actually the true axonal network looks like when we actually try to model with diffusion tractography? So there's different ways to visualize the networks. So there's in the human brain we can use Klinger dissection, where you have a formally fixated tissue. You freeze it and thaw and freeze and thaw, and then you very gently can delineate the major fiber pathways of the brain, like illustrated here. And this spatial, how it looks like on this one, you can actually compare it with your diffusion tractography and see that you get something which very much corresponds to these tracks that you extracted with Klinger dissection. But this is only the major pathways. If we want to look into more specific of the different axonal projections, then we need to go to our animal experiments where we can use invasive tracers. And this is a very nice work by Sharman and Pandya in the book, where they have collected different injection sites and visualized on the same template how they project in the brain. And this gives a lot of information of how can we actually expect how the tractography looks like. So we can see here from an injection site that we have a cord of axons going out here and then they spread out. And we can see it's a pretty complex network, even there's only a few injection sites visualized here. But very interesting is in this region here, the SLF is a very broad area here where you can see there's a pretty low density of axons So this really suggests that even if we have this large track, then it appears that it intermingles with other of these, the actions from one projection intermingles when it enters the major highways of the brain. So when we look into more detail, so there was specific tracks or connections we were looking with the tracers, but we also looked to of the microstructural features of brain tissue. So if we cut across here, and then we can look at the axon, how it looks like cross-sectionally, the axon diameters and its densities, we can see there's a high density of axons here. And that was in contrast to the BDA labeling I showed you before in the SLF, while appearing a lower density. But that's really not true. There's a high density of axons. So that's really indicate that these axons intermingle with each other when they enter these highways, as said. We can see it has been shown in corpus callosum that we have different axon diameters and distribution across corpus callosum. So that really means that there's not the same connection from A to B. It means something. And actually have been shown that these different sizes for axon diameters really correlate linearly with the propagation speed of the signaling going from along the axons. So the larger axons, the faster conduction speed we have of the signal propagating from A to B in the brain. And the mean axon diameters here in the brain, measured with electron microscopy, is below 1 micrometer. But we have a few of the very large axons. They are not really seen often. So actually, that the white matter that we are mapping with tractography has a physiological meaning. So it's not just a connection from A to B. It actually has a conduction speed and delays in the brain. So actually, the meaning of white matter is really to ensure the signals, how the signals are communicating in the brain, and ensure the function in how we actually work in the daily. And that has been shown an example here with Giorgio Innocenti and his colleagues here. They have been injecting in the brain different tracers, and then they have looked at the distance from these tracers and look at the axon diameters, and then they can actually start to map out how this physiological meaning of white matter looks like. And they have shown that there are different conduction speeds and delays dependent on the target of the region that a tract is approaching. And for example, you can read more about this, but generally the message is that motor pathways and contralateral projections are among the fastest connections in the brain. But in tractography, we really not can see this today. So today in tractography, we look at brain connections. So it's like a connection from A to B. So it's like binary. So we segment out the tracks. This could be like here, human work here. And we can compare this to tracer studies, because we also can see from tracer studies how it's like the projection, the spatial projection of these tracks in the monkey, for validation. We do try to make some quantitative measurements expressing the connectivity strengths of the confidence simply by counting the number of streamlines when we do tractography, but I will come back to that. So this was insight into what is actually the white matter that we are trying to probe. So now we go into this black box of tractography. How does this actually look like? So we can define tractography. So we can say it's a framework to non-invasively estimate and visualize fiber pathways in the brain. And we have an input as a subject here, often alive. Then we have our black box here when the data sets come out. And then we can have two types of tractography that we actually can visualize as deterministic and probabilistic. So let's have a look into the pipeline. If we first look into the deterministic tractography pipeline, then it consists of these three processing stages. And this morning you have heard about something about the acquisition. So in this one is the selecting the B values, the scanners, the sequence, but it also includes all the post-processing and ensuring the data quality. Then we have the fiber reconstruction stage where we extract the fiber directions. And then we have the last stage in deterministic tractography, that is like the fiber tracking, where we draw the streamlines. So the main part, or the main idea in this fiber tracking idea is that we have this local field of principal directions that we extracted from the fiber reconstruction processing stage. Then we select a region, our seed region, and then we start to draw a streamline. And that we simply do by starting here in the point in the middle of the voxel, as illustrated here. We go a small step in the direction pointing in that area. Then after a step, then we look at the local neighborhood and what is like the direction now. And then we update our direction, walk a new step in that direction, and then we continue to some kind of stopping criteria. So as you can see, the name of deterministic, it simply says that if you repeat this process many times from the same seed region and from the same position, then you will get exactly the same streamline position throughout the brain. So it does not add any uncertainty, or it does not add any probabilistic inference about the tracking you're doing. But that we can introduce if you go to probabilistic tractography. So again, when we look into the pipeline of the probabilistic tractography, we have some of the same processing stages as before in the deterministic. So we have our scanning and the fiber reconstruction, which is absolutely the same as before. But now we have included a fiber-oriented uncertainty, also what we have been, Flavio was talking about, the ODF. And then we have our tracking stage now, which is basically the same, but it's a bit different from deterministic. So the main idea of this fiber-oriented uncertainty stage is actually that in each of these voxels, we look at the principal direction, and then we try to infer how sure are we to walk along this direction. And then we create this fiber-oriented distribution saying, giving this expression for this voxel in each direction. So if we use this information in tractography, then the point is that we're going to have our principal directions. We start in a seed in the middle of the voxel, as illustrated here. Then we need to go in a direction. And then we look into, I need to go in this direction. And then I look at this voxel, go and sample for this uncertainty, but describe how sure am I going to this direction. So I add a bit uncertainty, extract it for this fiber-oriented uncertainty distribution, and then update my step in that along that direction, go a small step size in that long direction. Then a new position, look at the direction, look at the uncertainty, and then I sample for this uncertainty to add to the principal direction. And that's how I draw a streamline with probabilistic tractography. So you can see if I repeat this many times from the same seed region with probabilistic tractography, then I will start to see, I will not have the same streamlines projection throughout the track. So now I start to see something about how sure I am sampling this tract, giving this different information if, in each voxel. So how do I actually then get the probabilistic mapping? So you can have this probabilistic index of connectivity, which is PICO, this naming was introduced by Jeff Parker. So the point is that you simply take in each voxel number of the count, summarize the number of streamlines in each of these, and then you can add it here in this color coding. So the color coding is just fancy. So the main point here is that you have a seed region here. This is a broad seed region here where you have the highest intensities, and that's of course because it was here where you seeded most streamlines. And then with distance, you have fewer streamlines that it's describing. And the index here, we can have a voxel, and each voxel describe how well connected am I to this seed region. So you cannot say from the seed region or from this probabilistic index, even that this looks like a track, you cannot say really that this is how these are connected together. Then you need to inspect your streamlines. But this PICO index says something about how well connected am I to the seed. Okay, so that was like, we have these two last stages here that we have not been talking about today. So I will just go quickly into that. So this is how we actually draw streamlines. So I said that this is like the same procedures that you can use. And there basically exist two kinds of procedures. So we have the fiber assemblement by continuous tracking. FACT is shortened as, and this was introduced by Susumu Mori in the first papers of tractography. And the basic principle is here that you have your principal directions here. You start in here in your seed region. You take a step until you cross the boundary of the voxel, and then you update your direction in this voxel, giving the principal direction here. and then you walk until you reach the next boundary. So every time you reach a boundary, then you update your direction. So in this fact method, that you have different step sizes depending on the voxel size and how you protect through this grid. So you can say one of the, it's a fast and it's relatively reliable, but you have some problems if you have very sharp curvatures using this method. So you can go to another method. This is like interpolated streamline tractography, which is probably the most used in different toolboxes today. So the principle is here that we have this grid of our voxels with the principal directions, and then what we're doing here is that we actually interpolate this directional the directions, and then we have this high resolution interpolated principal directions. Then we start again from our seed region, and we walk in small steps and update our new directional from this interpolated directional space. So this is something that works on subvoxels, and there's many different methods that are implemented using this kind of interpolated streamlining. And many methods are using that. So we have briefly been talking about these different sources, ambiguities that Alex was talking about today. So and I'm trying to map out this. So through the tractography type pipeline here, given the different stages, then I try to map out how the different sources of ambiguity actually accumulate throughout the tractography pipeline. So, and I really believe that in the scanning is absolutely, scanning and post-processing is the most crucial part of the sources that you can introduce in your tracking. Then of course you have the fiber reconstruction, the matter of multiple crossing, multiple fibers or single fibers. Then you have something about the uncertainty, and then again you have your seeding strategies. And here I will focus on the fiber reconstruction and the tracking. So again, let's go back to our real-life tracer information, because that really can tell us something about our challenges there. And I have here played a grid. The voxel size here is relative to what one would expect to see in a clinical scanners. And we can clearly see there's very few situations or few voxels in this region here where you have single voxels. That's mostly complicated fiber configurations here. And that's we need to handle. And also, that's been seen earlier, the slide that Tim Behrens has shown that if you really do tracking with a single fiber model, then there will be the smaller association fibers that will be very challenging to extract, because actually you cannot extract it from these 9 subjects. And if you take it this short association fiber, then you will actually be able to track it with a multi-fiber reconstruction. So that's really suggests that the multi-fiber adds something, more information that the single fiber does not in terms of stratigraphy. But there are actually a situation where the DTI, which is the single fiber, most often used, that will work. And that's, for example, if you have a straight fiber situation in, for example, the corticospinal tract. So this is an example where we just have drawn a small seed here, and then you have a DTI, and then you can see the streamlining here. This is the deterministic tractography. And then you can apply, do the same thing for a data set where we have reconstructed for a multi-fiber, the constrained spherical deconvolution, and we can see more or less the same projection sites here. And that's because the corticospinal tract is a very dominating track projecting through, even though we have crossing fibers here. So the DTI more or less will survive and provide something which makes sense. But there's more complex fiber configuration. For example, very known is the callosal fibers here. So if you now place a seed here in the middle of corpus callosum and track here with a single fiber and the multi-fiber, well, this is still deterministic, then we see this well-known U shape. It's not because it's a wrong track, it's still a true track, but it's not the full truth that we're seeing here, because deterministic has a problem in these areas of central semivert where we have a lot of crossing fibers appearing. And that is not really what DTI is happy of following. So, but if we apply the multi-fiber, then we see that we do get the same U shape, but we also see that we start to get some more out to the lateral projections here, but it's not really convincing with this deterministic tractography. So if we still now use the deterministic here, as I showed you before, but we now use a probabilistic that allows us to sample the track with many more streamlines, then we actually could see the more true or expected projections that we would expect from seeding the mid part of corpus callosum to different parts in cortex here. So one could say the take-home message is here that for tractography, multi-fiber reconstruction methods should always be preferred. And how well you sample your pathways also depend on the method you're using and your seeding strategies. So when we set up tractography, there are some general settings. So there are some stopping criteria. When should we stop projecting a streamline? And that has FA values where it's really low FA values you should use. But as you can see the FA map, the problem here is that the FA is based on a single fiber model. And if we use a multi-fiber model, then a single fiber model should constrain a more advanced model. So that's not really optimal. So I suggest that you should try to avoid using FA values as a stopping criteria. So we have the maximum curvature. That's ensuring that the streamline are not doing a very strange curvature within the voxel. And then we, of course, have the brain mask, and that what would you expect, for example, in cortical regions, that we expect streamlines to terminate there, which is natural when we know the axons are projecting for cortical regions. Then we also have the streamline rejections in our method, and that could be different toolboxes for different things. That could be, for example, streamline length, so we can add a constraint saying we only want streamlines that have a certain length. If they're shorter than that one, then exclude it from our tract. We can also have streamline looping, and that could be this example here that the streamline loops around. So it does fulfill the curvature constraint, but it's simple in doing a very unplausible anatomical projection looping back. And that is something that when you inspect your streamlines, you actually can see this happen sometimes. So it's really suggested if it's offered, then add this check in the tractography toolbox. Then there are some other parameters to be set. That's, for example, the number of streamlines. That's always a thing that have been a lot of questions: how many streamlines should I use? And it's a really hard question to say because it's very much dependent on the toolbox you're using. So what I can only say is that you should really look into your toolbox and look how does it actually define, how does it submit this number of streamlines from a voxel. Some are putting out in the middle of the voxel, some are randomly selecting a position in a voxel or a region in the seed region and submit the streamlines. Then we have the step size, as I said before. So you just need to ensure it to be short enough, 1/5 or 1/10 standard. You can go very short, but then you just waste time of nothing. And then, of course, you can have select the tracking method, but often the different toolboxes always have selected that for you. So the take-home message here: check your toolbox, how it works, and the default settings. So now we enter something which has really important and influence what track system you're actually able to extract. And this is like the seeding strategies. So there exists many ways of doing seeding strategies, and as I said, it has a huge impact what you're doing. So I'll just go through here some of what I think is like the things which is general for the different toolboxes. So we have in the first one here, that could be free tracking. So that is tracking when there's no other kind of constraints. So we select a seed region, for example, the middle of corpus callosum here, and then we submit a streamline. And you see here that we go to both sides. And that's simply because the diffusion is bidirectional. So we cannot say if the streamline should go one or the other direction. So therefore, the streamline always projects both ways, just like that you know that. Then we can add some constraints. So we can say that I want this target region. This is the region where I want to do my statistic analysis, where I do want to investigate. And often this is combined with a termination region, because otherwise the streamline will just continue and can jump to different local cortical regions if its cortex this target is placed. But then this looks like this one, and still we have a projection bidirectional directions. Then we can constrain even more if I want a specific track. So I can use a waypoint. So I only want to have those streamlines that are projecting through this specific waypoint. and are still interested in what is happening in this target region. Now it could be that some of the streamlines that are looking even in this waypoint are not really the tracks I'm interested in. I do want to add some more constraints on the tracking. Then I can add a exclusive ROI in this area. So I only want to have this U-shape track. So then I exclude all streamlines that fulfill all criteria, but enter this exclusive ROI. So this is the different things that you could do to constrain Tractography. So these ways up here, that's mostly used for probabilistic Tractography, but you can also do whole brain deterministic Tractography. Is like where you like a seed in each voxel in the brain, and then you create like a database of all your streamlines, and then you can add the same seeds up here, but they are just called some logical numbers like and or not, and then you can actually filter out your tracks in another way. So that could be another seeding strategy, but that depends on the method that you're using. So the take-home message here, when delineating a pathway by constraining Tractography, it's really important that the ROIs that you're using, the waypoint ROIs, are not the ROIs that impact the shape of the track. So if you have, for example, a broad track, and then I want to constrain somehow, then it's important that your waypoint does not really is smaller than the cross-sectional area of the track, because then you start to infer what you think is a true track, but we actually want to have Tractography to show us the true track, right? So it's important that you are very self-critical and really are aware of these very tempted situations. So let's look at the potential limitation of Tractography. So as I said before, we have these different sources of ambiguity that are accumulated throughout the Tractography. And that challenges when we like to get insight into Tractography and its limitations. So we can actually just replace this with post-mortem imaging, and then we lower this major impact on the scanning session. And then we now look more at the methodological, the modeling parts, what is like the chancing here. And this also allows us to use our experimental animals here, because now we can use tracers as like a ground truth, some independent anatomical data that allow us to compare with our Tractography results. And that we did. When this next slide. Oh, that was too much. Oh, that was right. Okay, so this is just an example how the data set when you're using ex-vivo imaging. So I will not say much about this because there will be a talk later on this topic. But just say this is like a typical clinical in vivo in 10 minutes. But we can have this fixed tissue where I can go to very high resolution and very nice anatomical details, how things looks like, where you can just look at this color coding. And then you can actually start to manipulate or try to set up situations that is not possible on clinical scanners today, and see how your Tractography method actually works on these different settings. So we did some years ago a validation study of Tractography. And this is like the basis what I'm going to go through now. So what we did here, we used a pig brain here. So we injected a double tracer here, manganese, which is an MR tracer that is seen hyper intense on T1 weighted images. And then we have a classical BDA tracer, which is a historical tracer that you stain afterwards. Then from this one, we can identify the seed region. And then before we were cutting the brain for the BDA tracer, then we of course acquired high quality ex-vivo data set. And this was the data set we were using, applying Tractography to. So with the data set here, when we do this kind of validation, it's important that the challenges in Tractography in the data set, even that we can go to high resolution data set, but we selected an image resolution that is comparable to the challenges that we observe in vivo. And here, this is just showing the manganese tracer, where we can see the target and the projection areas, and this is just comparing how the Tractography looks like in ex-vivo dataset. So what can we learn for that one? So actually, we have 3 different brains here with 3 different injection sites, but I'm looking only on the prefrontal cortex injection, and we found different robust projections. So we got a contralateral cortical projection, which is a red, And then a cortical nigral projection, the yellow, and a cortical thalamic projection, what would you expect from tracers. And now the interesting thing is how is it performing on tractography? So we did see some false negatives not existent, what we expect to exist, and we also see a situation where we see a track, but it was not really the right one, for example in thalamus, where it's just skirt looping around here, and we can reproduce this false positive in other situations. We also see termination challenges here that we expect to terminate down here substance nigra, but the tracks just skirt around and continue. And the interesting part is also that when they continue, they just continue and stop suddenly in white matter without really approaching a gray matter region. And that is something I will talk about in the next minutes. But the main message of this study is really that the thing is that if you take hundreds of subjects and do the tracking on it, and you can find a robust brain network, you cannot really say that this is a true network just because that the tractography is able to reproduce it across many subjects. What you learn from this, a lot of subjects, is you just learn that how robust your method is. But as said, you can use another tractography method and probably it will show something different. So to be absolutely sure if your tracks are right, then you need to have some independent data to verify the tracking that you actually extract. But first, let's have a look at the termination problems. What can actually be the cause happening here? So we have different termination problems in tractography, and today there's a lot of discussion of this sulcus here, and this is mainly because that you have a very high dense fibers of U fibers that's making the projection from the deeper white matter to project into this challenging. We also have a situation in the gray matter structures down here that's very hard to project up here if you start in the cortical regions. So what can the problem actually be? So the example could be here that we have our image grid here, and then we have this smaller gray matter structure down here where it's low anisotropy. Then we have our true axons going that direction and terminate down here. But because, as I said in the beginning, we have a really high density of axons in the brain, then we of course have a major pathways surrounding this pathway projection to here that go around here and just continue to other projection sites. So if I take my seed and place it here, and try to reach this target, what I would expect, then in the beginning the streamline will probably try to approach the gray matter structure, but because this structure is like very heavily partial volume affected, then it will just follow the main anisotropy of the surrounding axon and just skirt around and continue. So we have a false, a true positive, and then it ends up in a false positive track. So partial volume effect is really a huge challenge in tractography, and you need to be aware of that. And that's both for deterministic and probabilistic tractography. But there's actually some tricks we can use here, because we know that the tractography is bidirectional, so we can actually set the seed there in the gray matter region that we expect, and then we can go in that direction. And that's many studies have been doing that, and you just need to be aware of that it doesn't matter where you're going from cortex to a gray matter structure, a deep gray matter structure, or a deep gray matter structure to cortex. So you might actually get something doing in the other way around. So then I selected to talk about path length dependency. So this is an upcoming problem. It has been there for some years, but now it seems that it has been making more seriously. And it's actually something which is related to probabilistic tractography. And the first really start to commenting this path length dependency that you can observe with tractography was actually Jeff Parker and Daniel Alexander in an early paper. So they have a huge seed region here up in motor cortex, and then you can actually see the probabilistic index here in the different parts. And when you then start to thresholding your probabilistic index, then you can start seeing that when you increase in the threshold, then you start to lose some of the longer tracks, and you only will get more contrast to the shorter tracks. You can also see here, this is the cortical spinal tract, and it appears in this subject that you have like a weird connectivity down here in the lower part of the brain, which absolutely is not true. This is a methodological problem, simply because tractography sent out streamlines up here, and only a few streamlines actually survive these longer distances, and then of course you have a lower connectivity hit rate in these regions. So, of course, we also have path length dependency when we do tracers. So we can have this is our grid, and then we have 3 projections here, and this is our axons. So in the beginning we have 12 axons in this voxel, and then we have 9 because they start splitting out, and then they split out, then they have 3, 3, and 5 down here, and continue with 5, and this is 5 all that way. So of course we have a kind of path length dependency also with tracer studies. But the main thing is that we have, when we are measuring here at diffusion, we are measuring the local anisotropy from diffusion, and we're sending out these streamlines by this probabilistic way with some uncertainty, and then we will lose our streamlines because they will bend off here along this track, will sample the track, and then even that we should expect like have a straight connectivity index here in these fibers, then because of this problem with the probabilistic tractography, you will still have a decreasing confidence. And then imagine if we get it more complicated. Now we have a crossing fiber situation here. Then we have our model, even if we can model our crossing fibers, then tractography gets even more confused. Because they start to spend a lot of streamlines trying to send out this more complex region, and it will cost streamlines going to these regions until it reach these more straight fibers. So actually we showed this in an example here. So we did this with the tracer I've shown you before. This is our popular pig brain here. So what we did was we have our tracer, so we know actually the ground truth. So we're looking at the prefrontal cortical projections, and we generated a canonical streamline, and then along that tract, the canonical streamline, then we counted the number of streamlines hitting this tract along the distance from the seed region and down here to this region here. So out here we have the number of voxels from the seed, which is the red striped line here. And what we would expect is actually that we will have a continuous heat rate of streamlines crossing this canonical streamline. That should not be dependent on distance, like the green one here. It should go up and down here. But what we observe with probabilistic tractography, this is the blue line. So I start in the beginning, just up here with the prefrontal, and then suddenly the heat rate drastically went low, very low, and then it stayed down here, where we come to the more straight part of the canonical streamline. And this is simple because as in this region up here, we have this very complex fiber, this centrum simovair. We have a crossing fiber, seen that many times today. And in that region, this tractography method spent a lot of energy of the streamlines to send out to really probe the track that it should follow. And therefore, you have a decreasing, very significant decrease in your confidence map, exactly as shown in the human data before. Now it has been suggested in some that this path length dependency can be like corrected with a linear correction factor. So the number of voxels you are going from the seed, then you can just add that voxels factor to the count rate. But if you do that, that's like the yellow. If you can see the yellow, then the yellow follows exactly the same problems as the blue one. So it does correct something down here, but it doesn't really correct this one. And that's simply because this path length dependency is a pretty much complicated and nonlinear effect inherited in tractography, and it will be different between different tractography methods. So how did we actually generate this streamline with the green one? That's a method we have suggested called IST. That's a iterative confidence enhancement for tractography. And this is simply a wrapper that you wrap around a probabilistic tractography method. It's like a region growing that can solve for this one. So one cannot say this is the true solution, but it allows us to have like an impact, and hopefully it will open for discussions, different ways to solve this problem. So, take-home message is here that the PLD effect is a nonlinear effect in probabilistic tractography, and it will affect the longest distances over the shorter one. And I absolutely think this is something that we need to go more into to really understand it. So, I want to make a general comment on this seeding strategies. So, there's different ways of doing seedings I've been told you today. So, we have like one could be that you seed in each voxel. And then you can like filter out your tracking. That is will not really be much affected by path length dependency. Or you can do probabilistic tractography where you seed in a region A here, for example, and then you look at the connectivity to C and B. And of course, the longer distances here will be more affected probably by path length dependency. So how you actually select your seeding strategies actually influence also if you want to do network analysis. And this is something that you can just read about Lee at their work down here. Okay, and the last thing is I will give a short example here. So how you can actually combine tractography in a new kind of way. So I will give an example of multiple sclerosis. So multiple sclerosis is a neurodegenerative disease, also dominated by inflammation. So you will start some inflammation in the beginning, then you have demyelination, and with time the neurons will degenerate, and you will see atrophy in the brain. So the challenges here that on the clinical structural T1 images, you see these hyper intense regions where you like this is the main attack of the MS, and you can also see on the T1 weighted images that you have hypo effect, and that somehow indicates that you have scar tissue here where the tracks are actually totally degenerated in these regions here. But the chances is actually that even that you can see this lesion, you can have the lesion node and the locations, then it poorly correlates with clinical scores. So how can we actually do that? So one of the things is that one can actually hypothesize that these lesions, which is like the local attacks on the tract network, so if you have different subjects here and you try to do a voxel-wise analysis like from DTI, where you have local FA values, and you have different attacks along the same track, then when you do a group analysis, then you will not really capture this attack on this group of subjects because the lesions or the local attacks are placed different places along the same system. But if we now use tractography here, then allow us to, it's sensitive to the specific track systems, even that these attacks could be placed different places. And one thing to do that is like using tractography in a new way. So we have the classical probabilistic here. But if we now take this one and do probabilistic tractography in each voxel of the brain, like this one, and then we got a lot of number of streamlines, and then we in each voxel count the number of streamlines from each of these seed. Then we get a voxel-wise map, ASM map, like this one. And the intensity here describes how many voxels is actually, this one actually describes how well connected is this voxel in a track system. But because that each voxel we seed is actually a summation of all the tracks in the network, then it actually describes something about its local network in the brain. So we did that, use this ASM and compared with the MS patient at different phenotypes. So we have the secondary progressive and relapsing emitting. And when we look at these two, it actually allowed us to differentiate using this ASM map. So we can see here in the coronal, we look at the coronal tracks, we get some significant points here, and we could not find anything with the FA map. So I have to say, if you look more and do more investigation here, it's not that the ACM is like a wonder magic number that will really solve all the problems that the FA does not provide us, but it will increase the sensitivity because it's more sensitive to the tracks system that FA does not measure. So the take-home message of this one is actually that it could sometimes be a good idea to combine tractography more with the FA values. And it's even the same dataset. You already have the dataset. So in summary, so to be a good tractographer, it's really a matter of training and experience. But you need to know your method. So one of the chances is here that the true axons intermingle on the major fiber pathways. So it's really hard to differentiate that with tractography. There's no guarantee that you can do that due to limited resolution. You need to know insight into your tractography method. You have the sources of different ambiguities here. So you need to be aware of that. So always ensure the quality of your data and always inspect the data. Then, of course, the selected seeding strategies you select has a huge impact on your results. So when you're publishing your stuff, I really ask you always to really in detail describe the seeding strategies that you actually selected. And when you compare with other studies, really look at the seeding strategies and see if this could be an explanation that you have a difference. Then, of course, tractography is a unique method. It's really cool. It's the only non-invasive tracer. So enjoy its huge potential, but you need also somehow to be realistic of its potentials and limitations. And always when you try and find some new findings, try to see in the literature if you can use independent data, for example, that could be TMS or tracer knowledge, to support your finding of your results. And then, of course, I will thank everybody, and Flavio for inviting, and those have been helping with the illustrations of this talk. Thank you.
Thank you, Tim. I think we have time for a couple of questions. If I'm sure there will be something about tractography you still want to ask, please don't be shy now. I know it's also time for lunch, but... Okay, one question coming.
Thank you. I'm wondering if you can comment on some of these recent papers arguing that tractography is inherently limited relative to tracer methods. Do you have any sense that with some of these more advanced methods like global tractography that maybe some of those limitations can be overcome, or do you ascribe to that viewpoint that it's going to be inherently limited?
Yeah, I think it's a good point. So I just covered here the general way of doing tractography. So I think that tractography is challenges due of image resolution and the sequence and whatever you're using. But one thing is really the missing part of tractography is that you have so much local neighborhood information that actually can infer when you draw the streamlining. And that's not only when you reconstruct the fibers, but it's also when you draw your tracking. On the global tractography methods, like try to adjust this in a way to look at the neighborhood or something, how the shape looks like by a track. And I think that's actually been, by Dan Alexander's group, has been coming out a work which is very nice from a seed region. You send out many streamlines, try to sample the fanning of the fibers, and that from that one try to decide in a local neighborhood how should the streamline actually project. So I think there's a lot of opportunities still to improve tractography. So I think it can be used. You just need to be aware of its limitations, and it will always be improved.
Okay, thank you. If there are no other questions, see no questions. So I think we can close here this session for this morning. Thank you again, Tim, and see you at everyone at 1 p.m. so we can start. Okay.